Hello, I am Jean Dodson-Peterson, and I was lucky enough to have Andy Walker as my major professor for my master's and my PhD work at UC Davis. It was actually during my undergraduate, though, that I first met Andy. And at that time, I was convinced I wanted to be a winemaker. It was when I was in his 101A and 101B courses that I quickly realized that I wanted to pursue the viticulture side of the industry. It was really his passion for amphilography, for grape breeding, rootstocks, and most importantly, his students that really led to that switch. When I'm asked how Andy influenced the direction my life has taken, I can, without any hesitation, say he is most certainly responsible for where I'm at today. He gave me the opportunity to work as a teaching assistant. He gave me the opportunity to have research experience. And during those years, I watched him share his passion for grapes and inspire the next generation of viticulturists and winemakers and researchers in such a way that I knew I wanted to be a part of that. He has this innate ability to draw people in and to inspire them to keep asking questions, to make learning a lifelong feat. In my own experience, he really pushed me to the boundary of what I thought I was capable of, and then gave me the courage to take that next leap to address the next challenge. It's because of him that I'm a professor of viticulture today. His advice and direction, though, has always focused on what would bring my life happiness. And I think that's true for many of his students. To this day, I'm absolutely honored to be the recipient of his frequent encouragement and, most importantly, his friendship. So thank you, Andy, for everything you've done for me and for the countless students that have come before and after me. We are all so grateful for what you've done, and we are wishing you the most wonderful retirement.
Hi, my name is Andrew Waterhouse, Director of the Robert Mondavi Institute and Professor of Enology. Welcome to this evening's forum program. As a public university, we pursue a mission of excellence in research and education. We have a mandate to disseminate knowledge and to foster a dialogue with the public through events such as this one. Forum engages the public with an array of scientific and research advancements in the disciplines of wine, brewing, and food to highlight the value of UC Davis research to the California and the global economy. Tonight's program highlights not just the research, but the skilled and creative personnel who gain valuable skills by studying with professors here at UC Davis. I want to mention that tonight's program is being recorded and will be posted on the RMI website next week, so please notify your friends who could not attend. I would like to thank those of you who submitted questions during registration. You can continue to ask questions using the Q&A tool, and we will get to as many of your questions as possible at the end of the program. Tonight's guest needs very little introduction. Andy Walker is a professor of viticulture and the Louise Rossi Endowed Professor in Viticulture and Enology. Without a doubt, he is one of the most famous grape breeders and geneticists in the world today. His research program has had significant impacts on the California and the global grape and wine industries, developing new rootstocks and grape varieties resistant to Pierce's disease. And as we will see tonight, he has mentored many young scientists who now hold important posts in industry and academia.
Thanks, Andy. Good seeing you.
I was hoping we could start tonight by having you reflect on how the technology of breeding, with respect to viticulture anyway, has evolved while you're over your career. I know that things have changed, and it would be interesting to see how things were different when you started compared to where they are now.
That's an interesting question. That's the tools have changed and they've evolved, but the purpose and the objectives have not. They're pretty much the same. And sometimes it makes you feel like you haven't made any progress if you look at it from that perspective alone. But it's tricky to judge that. Of course, the whole advance in molecular genetics, when I finished my bachelor's in 75 from Davis, it was in botany. And when I came back to school in 81, 82, in genetics and horticulture first, the world of genetics had changed completely in 6 or 7 years. And it has still on a trajectory that is almost limitless, it seems. Sometimes it's a bit scary, in fact, in terms of what it might be able to do. But it's greatly improved the way we can look at a number of traits. We can use all sorts of technologies that we hadn't had before. And the whole revolution in genetic mapping and markers has really dramatically improved the efficiency of breeding. Maybe not to change the objective, maybe not develop a lot of products oftentimes, but certainly achieved efficiency goals and acceleration goals.
One of the things that students comment on is going with you on these collection trips. And I remember hearing stories from Olmo about traveling the world and collecting vines from Afghanistan and Pakistan and other places, which I think might not be the safest place to go these days. But I was going to ask you about your collection trips. I know you've done quite a few. What was the purpose for those?
So those trips were inspired by Harold Olmo, and if you haven't read his oral history from the Bancroft Library in Berkeley, you should do that. It's just phenomenal adventure stories, and we call him Indiana Jones of viticulture because of that. And of course, I wanted to emulate that to some degree. But when I asked my wife if I could go to Afghanistan or even Pakistan, she said absolutely not. So it's limited what I had to do. But when I started my career, we had one accession, one individual of Vitis arizonica in our collections. And this is the southwestern species, just one plant. If you looked at the number of other species we had, there were 20s and 30s of those to sort of explore the diversity within them. But one given individual. So I said, well, I have to do something about that. And the first was understanding what it was. And it turns out that the southwestern United States and northern, western, and central Mexico are tremendous hotbeds of Vitis. Lots of speciation going on, lots of hybrid zones, absolutely fantastic place to collect. And it's a nice place to collect. It's quite easy. All you have to do is look at a topo map, and where there's permanent water, you'll find grapes. And that permanent water can be a little spring, or it can be and as long as it's not an ephemeral stream, the grapes will be there. And that's because the birds are there. And the birds are the primary driving evolutionary force in viticulture that we just sort of ignore. They're the reason grapes are small and black and seeded and grow up in trees, because that's where the birds are, right? So it's a very interesting phenomenon.
Well, I assume you were there also because of specific traits you were looking for.
Yeah, at first it was just sort of, let's get as much as we can. And then we stumbled onto a number of situations. It'd be worth having a whole separate forum about the origin of this group of materials that I have that Harold Olmo gave me as seeds. It's absolutely fantastic story that sort of came to light by study. And a lot of that study was done by Samara, who'll give you some feeling for it tonight. There's this diverse material that has potential to be PD resistant. We knew because of the locale where it was growing. We stumbled onto the fact that it has good nematode resistance, quite remarkable to some species. It also has great salt resistance and salt tolerance. It has the potential to be more drought tolerant, although that's tricky with grapes because I'm not sure it exists. That was one of the questions that came up earlier. So it had all these various characteristics that were truly amazing. And the only place we saw a lot of these characters clumped together, combined together in a given individual or given species was in rotundifolia, and we couldn't use rotundifolia in breeding because of sterility barriers. We couldn't actually make fertile hybrids. So it's an amazing story, and it unfolded quite interestingly, and has provided us with 20, 30 years worth of work, and there's another 40 or 50 to go.
So is drought resistance something that's difficult or problematic, or what's you sort of alluded to that?
Yeah, it's a very interesting problem, right? So if you think about grapes again, and the Southwest is a great place to think about it because they only grow where there's water. There's only one species of grape that grows without obvious water, and that grows in West Texas, way out in the horrible mesquite and juniper laden hills. No soil, no spring. But it adopted the very common ecological or environmental adaptation trait, which is if you're going to grow in a dry area, don't grow. And that's what most desert plants do. They grow with in micro amounts each year, very tiny development and improvement in terms of their girth and size, but they survive. And that doesn't exactly make a useful grape rootstock. So in terms of grape rootstocks, drought resistance comes from depth, roots that are able to plumb and meander through crevices and whatnot and get to deep pockets of water. But there has to be water there. So the drought resistance per se is one of those traits that's definitely not grape-like. And most vines are in the same category. They need water to grow because they have to grow above the tree canopy that they're supported by. Otherwise, they don't get any sun. So it all sort of fits together when you think about it functionally.
Certainly, one of the questions everyone is concerned about these days is climate change and how things are going to be different in the, I don't know, the next decade or the next 2 or 3 decades. And so I guess I'm asking, I don't know whether you want to answer this, what advice do you give the people who follow you about, what traits should they be worried about or trying to build into new materials?
I think climate change is going to be a revolutionary phenomenon in terms of grape breeding and grape development. And I always tell students in class in various places that viticulture is the most backward form of horticulture that exists. We haven't changed our plant material for hundreds of years, and we're proud of it. In fact, it's becoming even more restricted and reduced. We use very few cultivars, 10 to 20 are the vast majority of what we use on the planet. And there are over 3,000 of them. And they grow in areas that are incredibly hot and relatively dry across various desert areas as well, and colder areas and wetter areas. And there's tremendous variability in terms of aromatics and acidity and ripening profiles, things we can use to address the changing climate. And we're going to need to. We've invested around the world probably trillions of dollars in infrastructure to support wineries and the wine industry. If you think about all the chateaus and all the wineries and all the mega wineries, a huge amount of money. We're not going to lift them up and move them. We're going to have to change the varieties to adapt to the climate in those areas. And there are perfectly nice wines made. Those 3,000 varieties that exist somewhere on the planet, they make actually a fairly nice wine. Not everywhere necessarily, but somewhere. So we're going to have to rethink how we do that. And we can do that through breeding as quickly or probably more quickly than we could through going back and assessing those traits in the cultivated varieties.
So you think there's going to be a market for new varieties?
People are going to have to get used to it. And it's up to, I've told several people in very large winery operations that it's up to them to start this marketing process to get people comfortable with this stuff, not to reduce the diversity, but to in fact start increasing it and get them used to these concepts. And it's a wine style thing as well, right? You have to adapt to a new and different wine styles. People in wineries always tell you when you go to the tasting rooms, we can sell out all the Vermentino we can in the tasting room, but it doesn't compete at all in the supermarket or in restaurants. And so they don't produce it. They produce 50 cases a year and they sell it all in the tasting room. So that's a good example of a variety that does quite well in hotter areas and is quite aromatic, very pleasant, and really hasn't caught fire in any way, which is sort of too bad.
So do you have some other varieties you want to suggest that people be taking note of?
There was one of the questions here that was quite interesting, and it recalls a conversation I've been having with Randall Graham, the famous Rhone Ranger and Pinot devotee, about how to breed grapes, in essence. And the question always is, well, what should we use? What can we combine together and look and see what's there? That's the fun thing about grape breeding is you can actually do that. You can grow these populations of seedlings, and there's tremendous variation within those populations. All sorts of new things can be found and utilized. And I think there's a lot there, and it really is only suppressed by our imaginations in terms of what we want it to appear like or behave like. In general, if you look back through the parentage records, and we can now, through DNA marker technology, we can now go back and determine what the parents were of a given variety. And we're always very proud to think of these great noble varieties as being sacrosanct and, mount on the hill sort of thing, when in fact, they were crossed. Someone crossed two varieties together and made another one, and they selected them as well. And this didn't happen inadvertently. The petals and the pollination biology of the plant makes it extremely rare for it to happen by random or inadvertently. So someone created those processes, which means they've been studying grape biology for a very long time, because a lot of these things are hundreds, if not 1,000 years old, a lot of these varieties. So it's great. It's something we have to get used to, and we've just forgotten about how to use it. When I first started studying—
Wait. Are you saying that people have been breeding grapes that far back?
Sure. Why not? They've been building pyramids. That's much more complicated than grape breeding. I'm a little hoarse today because I've been out in the pollen all morning, and I'm quite allergic to grape now. But it's been going on for a very long time. It's nothing particularly exotic.
But are there records of this?
Nope. But only by the record of the pedigrees. So how do you explain that all these varieties that we use are in fact known hybrids of two other varieties without the fact that someone had to create them? It didn't happen just spontaneously. It may be very rare examples, but not spontaneously. And of course, grapes were being selected and bred by birds before the people were doing the same thing, right? They were directing crosses, they were moving seeds, and grapes were actually sort of migratory and animal-like in terms of their genetics oftentimes, because of the movement of the seed by birds. So you get a lot of things that grow in together from different areas. The wild species are very different than the cultivated types because they're male flowered or female flowered. And so it's impossible for those to self, and crossing occurs all the time. But generally it can be fairly tight within a group of materials, or at least within a region where those plants are surviving. But all these cultivars that we've seen and thought of as being sacrosanct all this time, most oftentimes, are known hybrids of two other varieties.
Well, the one that I think is the most obvious is Cabernet Sauvignon.
Mm-hmm.
But
Sauvignon Blanc and Cabernet Franc, and you wouldn't believe it unless you looked very closely at Cabernet Franc and Sauvignon Blanc in an amplographic sort of sense, and thought about what would the intermediate plant between those two be, and you realize suddenly, I can see that. And if you actually put in some of the other enological characteristics as well, pyrazines and these other things, at the same time, it all sort of pieces together.
But if you went and crossed Cab Franc and Sauvignon Blanc tomorrow, right? You wouldn't create Cabernet Sauvignon, would you? Or would you? You could. That's the tricky part. It's a matter of, you can't have exactly, but I think close enough so you'd be impressed. It goes a bit back to my new varieties. One of them is a dead ringer for Sauvignon Blanc. And we had a tasting a few months ago at Silverado Cellars, and they brought out their best Sauvignon Blanc, and they bought up a couple of other Sauvignon Blancs. And they had one from France that was $150 a bottle, and there was another $75 one from Napa. And they laid them out, and nobody could say that the wine that I brought with me as one of our new hybrids, Caminante, was not a Sauvignon Blanc. They said it very much like Sauvignon Blanc. It was indistinguishable in a tasting sort of sense. It's completely different than Sauvignon Blanc in every other way, and it shares some of the parentage, and that was Cabernet Sauvignon. So it's sort of a grandfather, grandmother, grandchildren relationship in a way. Really? Well, so when you were making these PD resistant varieties, I mean, how did you decide, what to cross with what in order to get a certain, or could you predict where you were going in terms of flavor profile? So that was the interesting thing. And when you ask me about what I would do in terms of grape breeding, the foremost thing in my mind is always resistance, disease resistance. We don't need any new varieties. We have 3,000, but we do need some of those examples with good, strong disease resistance. And we have that available now. The wild species have plenty of resistance. The question is, how do you get that resistance into a vinifera cultivar and leave all the obnoxious characteristics that are associated with the American species? And those are incredible acidities. Some of the riparias that we've tested have pHs below 2. It's phenomenal. And you can feel your enamel sort of dissolving when you're chewing on them. And the very aggressive peppery characteristics and high acidity, of course, and stringency, they're just not pleasant. So we knew when we started this process that we made that first cross, we call our first generation cross, that would be 50% vinifera and 50% Arizonica, say, or Arizonica rupestris sort of materials. When we crossed that together, that the
materials that we ended up with are not worth eating, let alone drinking. So our objective was then to take that and cross it back to a good vinifera in repeated cycles as quickly as possible. So when we do this, the first generation is 50%, of course, and the next is 50% vinifera, 50% wild species. The next cross to a good vinifera is 75% vinifera, 25% wild species, and then 88.5, and then 94%, and then 97%, and then you never can get to 100%, but you slowly get there. And we knew that we wanted to be in the high 80s and 90s before we started worrying about them. So we made a few wines at the lower levels of vinifera just for fun, just to see how they would look. And it was mostly Alan Tencher's desire to see those, and he was my assistant breeder, so working through that process, we got there. But the real question was, what are they like when they're much further along? And this is something I think that's a basic flaw with resistance breeding that's gone on so far, because people thought we've got to make wines and choose the best wine producing variety for each of these generations. But that's not true. You don't need it. What you need is something without the worst parts of that cross. So you exclude things, you don't include them in a way, and it makes it far faster, and it makes it achievable. If you spent your time making wine from each of those generations, you grow your seed up, which we can now go from seed to seed in 2 years in grape, which is very, very fast. So it used to be claimed to be a long cycle, but it's not. As long as you force it horticulturally, you can get it to do that. But you can't make wine for a long time, right? You have to take that one seedling, you have to replicate it, so you have a number of individuals, up to 10. You need enough to make a decent sized lot of wine, and then you need to make that in a reproducible fashion over years. So suddenly it's starting to take 5 to 15 years per cycle as opposed to 2. So we realized that it wasn't worth worrying about that. We did juice assays. ATS Labs was very generous over the years and did a lot of our testing for us. We were able to sort of define these things before we went through in terms of their wine quality without making wine. It was juice quality in essence. So that helped tremendously. It was a really big advantage for us.
So I take it the improvement in the technology of various technologies allowed you to move forward with much less time between generations, right? I remember hearing about this fellow, I think it was a USDA breeder down in the Fresno area. And during his career, he didn't produce a single marketable product because there was so much time in between generations.
Yep. That's a problem. And it depends on how you define your generations, right? So to what level you're going to select within that generation and what traits are you chasing after at the same time. So when I started this whole process, I went down to see Roger Bolton downstairs, and after I was on the faculty, and I said, Roger, I need to make wine at a 2-liter scale that would typify a 200-liter and a 200,000-liter scale. And I said, I need to be able to do that to select the best individuals in each of these generations. And he said, you can't do that. I said, what do you mean? I have to do it. He said, you can't do it. So in the end, we just ignored it. And we went through that last stage where we could make more wine and spend more time in those last few generations to really select and look for what we wanted, what we needed.
All right. We're going to move on now and bring in some of our other guests. So our next guest is Luigi Bavaresco, and there is Luigi. Hey, good evening.
Hi, Andy.
Or is it good night where you are?
Yes, it's night in Italy right now.
Yeah. So Luigi, can you tell us a little bit about your experience when you were here working with Dr. Walker?
Yes, good evening to everybody. And I wish to thank very much indeed the two Andys, Andy Waterhouse, the director of the Robert Mondavi Institute for Wine and Food Science, and Andy Walker, the protagonist of this forum, for inviting me. It is a real honor to be here today with you. And I am also glad for this event because it gives me the opportunity to go back around 30 years when I was over there, to feel a little bit younger than I am. So introduce a little bit myself. I am a professor of viticulture by the Catholic University at Piacenza. It is a small town located in the northern west part of Italy. It is quite young. This year we celebrate 100 years. And my main fields of investigations cover grape breeding for disease resistance, namely the mechanisms of resistance, and especially the phytolexin production, the resveratrol, viniferins, so on, and their rootstock physiology. And a decade ago, I was appointed director of the Italian Research Center for Viticulture at Conegliano for 4 years. So I left university for 4 years and my position was kept frozen. Then I came back. And during this period in Conegliano, I began a breeding program with the glera, the variety utilized to produce the Prosecco sparkling wine. I tried to get a glera-like disease-resistant new variety, and the works are in progress. And also over there, I was the responsible of the Italian grape repository, and it was released online at that time. And breeding is also a subject I deal with at the international level because I am the president of the expert group Genetic Resources and Vine Selection by OIV in Paris, the International Organization of Vine and Wine, which, you know, it is an intergovernmental body grouping almost nearly 50 countries all over the world. In the past, also USA used to be a member, but unfortunately left for some reasons. And it is a pity that USA is no longer in this important organization. We hope to get again on board also USA. But it was the rootstock subject to take me to UC Davis because I got my doctoral degree in France, in Montpellier, on Lyme-induced sclerosis, a subject where the rootstocks are very much involved. And I wanted to look for a postdoc. That's why I wrote in 1990 to Professor Mullins. It was the department chair asking for an internship. I had chosen UC Davis for the great reputation of this institution, of course. It's the #1 in the world. And Professor Mullins wrote me back, yes, you can come, and your mentor will be Andy Walker. So I was very happy. I right away applied in Italy for a scholarship. I got it by the Italian Research Council. And due to bureaucracy, I came to UC Davis in 1992. So in the meanwhile, Professor Mullins died. I had not the chance to meet him personally. And the first impact with UC Davis campus, I was there with my family, my wife Flavia, and my eldest son Enrico. The first impact was on Sunday, I remember, the powwow, Indian powwow in a building, maybe in the gym of the campus. And the following day, on Monday, Wixon Hall, where I met Andy Walker. And he addressed me a very warm welcome. And he explained me the subject of my research. You know, a research project, in a research project, there is the goal to be reached, the methodology to apply, and then you run the experiment. The goal was very particular. The goal was to find out a new method for establishing Xiphinema index in dual culture with grape. So I was a little bit scared about this. It was a challenging issue, and I began working with not good results. I was a little bit disappointed at the beginning, a little bit frustrated, but no problems. When you work with Andy Walker, you feel very comfortable. You feel motivated, even though an experiment doesn't succeed, because Andy will push you to retry, and he never gets angry and upset, and he's anytime willing to help you, demonstrating to trust you very much. So he is a mentor, a master, and over time also a friend. And I am also grateful to Andy for a great experience, exciting experience I had together with Andy, a trip looking for wild grapevines to collect germplasm in Mojave Desert and Death Valley. Very impressive, especially Death Valley, this terrible environment where In some places, the white grapevine was flourishing, green leaves, no drought problems. Very impressive, the behavior of this very fascinating plant. And then we collected the sample. They were shipped by plane to Sacramento. And I know that Andy took many other trips, scientific trips, looking for native germplasm all over states, maybe also abroad. And in this sense, he was in the footsteps of his predecessor, Professor Olm, as you already mentioned at the beginning of this forum. And he's in the footsteps not only for looking for wild grapevine, for germplasm, but also for the results. He got some new rootstocks and the new PD, Pierce disease resistant varieties. And another aspect I wish to emphasize is something after work. I appreciated very much the hospitality of Andy and his wife Viv when they invited the lab crew with the families at home for dinner, and, barbecue, good wine, good company. And this is a good way to build up a team spirit, which is important also during the work time. And I have many other things to tell, but I want to stop here because I want to keep some other aspect to the final statement. And thank you for your attention.
Luigi, can I interject just for one moment, in case you forget this story? Luigi was sleeping on one of these days on the Great Mojave journey as we went through the desert, and he suddenly woke up from the van and stirred, and he looked over my shoulder at the speedometer and at the mileage gauge, and he said in this awestruck voice, "My God, we've traveled the length of Italy."
Yes, a very long trip. Impressive. That's correct.
Thank you, Luigi.
Very welcome.
Thank you very much. Our next guest is Remy Cohen. Remy is not an academic. She's managing I would just say a wine company over in Napa Valley. And I'll let you continue the introduction, Remy.
Thank you, Andy. I'm so happy to be here with you and Andy Walker. This is such an amazing evening, so thank you so much. So as Dr. Waterhouse mentioned, I went over to the dark side, as Andy Walker likes to put it. So after getting my master's degree, working in his lab and as his teaching assistant with Andy, I moved into the industry here in Napa Valley shortly after receiving my master's degree. And I've been working as a viticulturist for the first half of my career, and then the second half of my career moved into management. I'm the CEO of Domain Carneros Winery. Some of you may have seen it driving by in Napa. You can't miss it on the highway between Napa and Sonoma. We specialize in producing sparkling wines and Pinot Noir in the Carneros region, where also my career began. But my first introduction to Andy is an interesting one. I went to UC Berkeley and studied genetics in my undergrad. And I took a year off to figure out what I wanted to do for grad school. And I was working in a genetics lab doing research on strawberry and grape. And I was researching also at the same time graduate school programs in plant biology. And I saw the program at UC Davis, and I knew right away that it was exactly what I wanted to do. And coincidentally, there was a man who worked at the genetics lab who was leaving to go to the program at UC Davis. And he said, "You have to call Andy Walker." And I said, okay. So I immediately called Andy Walker in minutes of even hearing that viticulture and enology was a career. And when Andy had heard that I went to Berkeley to study genetics, he was pretty excited about somebody who might be interested in genetics and would come and work in viticulture in his lab. So I will forever be grateful to you, Andy, because you offered me so many opportunities to help fund my degree, help make my degree easy in the process there, enjoyable and exciting through research and through being your teaching assistant. As being the TA for your 101 series, I felt like the most advantageous part of that was that I got to take the class twice. So I really got to have everything sit in and really learn everything that you were teaching because you just really espouse so much knowledge and so much information. And like I said, I'm so forever grateful to everything that you taught me and you contributed to the industry. And I have to say that even though I've went over to the dark side, that my favorite part of my day is walking in the vineyards and doing the work in the vineyards and learning about VitTech and staying abreast of what UC Davis is doing and what you're all contributing to the industry. So I wanted to take a few moments to let you know, Andy, that I think that you have all the best characteristics of a professor and also why you've been such a major contributor to the industry. First of all, it's obvious that you're brilliant. You have an exceptional body of work from grapevine rootstock breeding, all the new grape varieties you've created, the Pierce's disease resistance and research, which we clearly need in the industry. And we've seen a lot of loss and devastation from that in the last 5 or 6 years. So we thank you for all of your help and work in that regard. You're a very fast talker. You have so much to say, so you have to fit it all in. We only give you an hour a lot of times. And so you have to speak fast so that you can actually share all the knowledge that you have. And I always learn so much from you, even tonight. I feel like I've learned so much in the first half of this presentation. And it's so cool to think about the evolutionary biology component to our industry and what we do. You're always willing to help. Even all of us on the dark side, we call you all the time, and it's really not your role, but you'll still, out of your own curiosity and desire to learn, will come out and identify the rogue variety and the rogue rootstock and help us figure out what we're doing here, because we're always forever lost in learning in this industry. So thank you for all of your helps with diagnostics and making recommendations and helping improve the industry. You're obviously like incredibly hardworking. In fact, I don't really believe you're going to retire, but we'll see. We'll see. You're very genuine. You always are very authentic, and you always tell it like it is. And everyone really appreciates that about you. You're practical. In fact, I got my my habit of wearing hats in the vineyard from you and your Tilly hat. And most importantly, you're always smiling and you have a great sense of humor. And that is from really evident in both your ampelography descriptions and the fact that, like Luigi mentioned, your hospitality, inviting, your teammates, your lab mates and your lab and students over to your home to do white elephant gift parties and things like that and barbecues and really build the camaraderie in the department. And to that, we're very, very grateful. And with this, we say congratulations to you, Andy. This is a very well-deserved retirement, even if I'm somewhat dubious that you're really going to.
Thanks, Rem.
No funny stories there, Remy?
Just some. I have very fond memories of the white elephant parties at Andy's house with some funny gifts and some good humor. But I feel like Andy's always so good-natured. It's always a good time to be in Andy's company.
All right. Well, thank you very much, Remy.
Thank you for having me.
All right. We're going to move on. Our next guest is also from Europe. So it's not early evening over there. Astrid Fornick is a professor in Austria, and I'll let you do the rest of the introduction, Astrid.
Yeah. Hello, everybody. Hello, Andy, and dear colleagues, of course, on the screen. Very heartfelt greetings from Vienna at the Danube River in Austria. And from my side, it has been almost 30 years since I first met Andy. Then a very young and very, I would say, vigorous student from agronomy and viticulture at the University at Hohenheim. And I was looking for the best place to do research on grapes. And I've been sent by Professor Allerveld in the generation, I guess, from Professor Olmo. And he was so kind to recommend contacting his colleagues at UC Davis. And after, at that time, some kind of laconic fax messages that came to our department confirming basically it would be okay to come, I was basically the most proudest visiting scholar in the Walker Lab at Vixen Hall at all times. That's that I feel that way. Very, very special place. And with a remarkable community within the lab and all around. So Andy turned out to be, for me, an exceptional scientific tutor, a profound and challenging teacher, very challenging sometimes, a very kind-hearted coach, I would say, and a very having a very dry sense of humor, which is, I think, a very good thing to have if you are running a big lab and so many programs like that. I was then, from then on, before I didn't really know what to go for and what I could do, and didn't have the esteem what you need, but with Andy and the surrounding, it was all about genetics, viticulture, and phylloxera research, which after then has become far more than a job. It has become a passion, I think. And believe it or not, I got a project, and that was challenging and a little bit making me excited, but it was to create a dual culture of grapevine and phylloxera. I guess we heard that before a little bit. That was Luigi working with Xiphenema, so I did the phylloxera part with others. After graduation, I started to plan my PhD project that I wanted to target, and I figured Davis would be the best place to go, and Andy, of course, would be surely a very good advisor. So that time, no fax, but email. I used email and wrote an email. Is there any chance to follow up that work to Andy? And Andy said, sure. We are ready for you to come whenever you are ready, or we are ready to have you whenever you're ready to come. And that was basically the click when I knew that is the way to go. So he became one of the advisors of my PhD thesis, and I did most of the PhD work together with Andy and his lab in Davis. And I did student, it was so much to learn from him. The width and depth it takes to be a professor. Far from that, the lifelong passion, it needs to be a viticulturist. The patient, it needs to be a good scientist and to do science in the end. And well, in the end, how to differentiate among St. George and 3309, but this is another story. You remember the snake I was running from? No. At this credo, which he embodies in full, the most important thing that I still have in my mind today is that what really is important is that teaching must be fun, not only for the students, but mainly for the teachers. And I do embrace this. And no matter how research driven and media enhanced and controlled our academic system may work, it's still very important to me. In the end, and now I come to the clue, and you would not miss out showing up for my PhD defense back in Germany in presence, no Zoom then at that time. And after all been done and said, we started on a collection trip, guess where? Through half the continent, not looking for Vitis arizonica, but looking for real Vitis sylvestris. True, in the river banks of the Rhine Valley, when we crossed the country, we had even Peter with us. And we were driving with a very cute, small, chubby little smart cars. And that was really, really hard to draw. So another highlight, I'm running out of time, I'm quick, in our shared history was when I was able to spend a summer at UC Davis as a guest researcher and lecturer. And again, it was all about genetics and viticulture and phylloxera at the new Robert Mondavi Institute, It was all the same passion, same feelings. Only I had forgotten how hot that place could be. So fast forward, many years of joint research projects, papers, conferences, among the many things that I admire on Andy's being a tutor and friend is the consistency of his attitude from rookie researcher, student as myself, to being a teacher myself. He has never treated me differently, and I'm still inspired by him as the teacher of the teachers. And I would not be here without and having the chance to speak as a full professor and the head of the institute and doing so much things without having him tutoring and mentoring me as he did. Not only me, but he fanned and helped channel the scientific fire. I put that on here because I found it a nice picture of so many of us. So thank you very much, Andy.
Thank you. Thank you, Astrid.
No funny stories, Astrid?
Too many.
Too many.
I saved them for the last statement.
Okay.
Yeah.
She just has a remarkable skill to smuggle smokes, or we won't talk about where she smuggled them. But she's quite good at that.
No, I called him once and I said, they took my passport. What should I do? Remember? I don't say that details now. They were all frozen though. They were dead.
Safe.
Yeah.
All right, well, thank you, Astrid. We'll ask you more about smuggling later. Our next guest is Samira. Where'd Samira go?
Hello.
There she is. So Samira, I guess, came to Davis most recently, but she hasn't left. So I'll let you do the rest of the introduction, Samira.
Thank you so much, Andy, for giving me this opportunity to be part of Andy's legacy. So as I know Andy from my connection with him started in early 90s when I was a graduate student. And first he was my teacher, and I was taking Viticultural Practices 101, 101A, B, and C series. And in those classes, Andy packs a lot of information, as Rami mentioned, that he talked fast because he has to cover a lot of information and material, but Andy makes it effortless. So just moving from one cultivar to another cultivar, doing the amplography features, and all the stories which he tells in those classes that makes following from one cultivar to another cultivar more fun, and understanding and learning more fun also. I think pretty much all of his students, they know the story of Dead Bunny, which I think Andy is very good in narrating that story. He can tell us that himself. So after that, Andy was also in my qualifying exam committee, which is for all graduate students. That's like a one big milestone. So when I was super nervous and I went to his office and asked for the guidance, and he said, well, we are there not to fail you. We are there to pass the exam. And I think that was very helpful. So he was also part of my thesis committee, and he made it very easy to finish me my dissertation on timely manner. And after that, I needed a job. I said, okay, I went to Andy's office and I said, Andy, I needed a postdoc so I can continue. And I haven't finished my sentence. He said, well, you're hired. When can you start? And then rest is the history. So I joined his lab as a postgraduate researcher in 2002. So my time with Andy is 25 plus years. And my entire time in Davis, we have known each other. So in our journey together, and I think Andy alluded to the mystery of 89 population, the series of our investigative work, which we did. That was my first project, finding the Pierce disease resistance. And I started working on that genetic map. So when I started working with SSR markers, things are not like, nothing is coming from rotundifolia. And I said, well, Andy, I don't think this is a cross of rupestris by rotundifolia. He said, well, it has to be, because that's what Dr. Olmo, when he inherited that material from him. So we end up going back to the old Armstrong vineyard, which no longer exists, which is across the freeway where our new building is now. And we went and we looked at the mother vines of the Rupestris, Ediceras, and Wichita Refuge, and we took the DNA of all other grapevines which were growing, all the rows next to them. So that was our first investigative work and my other colleague from Italy, Silvia Vezzuli, she was doing part of her PhD research in Davis. So together we determined the parentage of all those 89 series, which Dr. Walker inherited from Olmo. And that brought the limelight on the Vitis arizonica, and we understood that how this material is very unique, and it has resistance to nematodes and phylloxera as well as to Pierce disease. So my other projects which we worked with was powdery mildew resistance, phylloxera, nematode resistance, and in last decade we moved toward the salt tolerance and the understanding drought tolerance. So the main focus of the reason for Andy's success as a grape breeder is that he understood the importance of germplasm. So he essentially took the baton from Dr. Olmo and from him Bioletti. So essentially between Dr. Olmo, Bioletti, and Dr. Walker, there is 100 years of history of germplasm collection and management and using that germplasm to do the grape breeding. So I had the opportunity to work with all of his graduate students. I mentored multiple generations of students and be involved. So Andy as a boss, we used to call him Big Cheese, and we are all small cheeses in the lab. So he is very compassionate, humble, and that is the most important part, that he is very accessible to all of his graduate students, all of his undergraduate, like whoever is working for him. He's really people person and take his time to build relationships. And that is for me, I hope I have got that imprint on me also, that positive quality of Andy. And I hope that in my future, in my career as a grape breeder, I will be imparting this knowledge to the next generation. And I also joined Andy on multiple collection trips. And I remember, I think it was 2011 or 12, right, when we went to New Mexico, Arizona trip. And so these collection trips are very brutal. They are starting like 6 in the morning, and we are driving 400 to 500 miles in a day. And they end around 7 or 8 p.m. And then we have to, like all the plant material which we have collected, we need to make sure that they are alive. And so, so in one of those collection trips, now we were driving for almost 3 or 4 hours, and so far, not a single grapevine in sight. It's like, okay. So in one of the ravine, we found, oh, there is a grapevine, like you could see it from a distance, that the grapevine is growing on top of other native plants over there. And lo and behold, there is a dead horse right there. Dead horse which is decaying, and you could not, like, the smell was really, really strong. But the grapevine was right there. So we needed to collect the samples. So, and it's like the graduate student, Claire Heinitz, she's working in USDA, and she's a curator of Violi there. So both of us, that was our first sample, and I have the picture, and I think I will always remember that story. That was also the trip when I first have a sighting of a rattlesnake. So I was just trying to mesmerize and trying to take picture, and Andy and Claire, they looked at me, Sumera, get away. He's just going to, he was rattling his snake. He was pretty agitated, but there were so many other sounds that it was hard for me to actually hear that if the snake was making a rattle sound. So there are lots of good memories, good times together, and I hope that I don't think Andy is, even after his retirement, he's going to be retired. I am looking forward to seeing him in the vineyard on his vehicle, and because we still have a lot of stuff in the pipeline. And the new person which the department is working with or going to hire, they have a very big shoes to fill. So I'm looking forward to working with Andy and get his mentorship, get his guidance wherever I needed the help in future. Thank you so much, Andy. You have been a wonderful, wonderful mentor in my entire career in Davis.
Thank you.
All right, thank you, Samara. We're going to move on to a few questions, Andy. So let's see. Here's a good one. How did you decide on the names of the new PD-resistant vines?
So it was done in sort of a comical mood. And if you'll go to Google Translate and type and get it all organized and type in those names, you'll see a pattern after a while. And I'll just leave it at that. And they're also a mixture of Spanish and French. So it's a Spanish surname and a French modifier, I guess, if you want to put it. Instead of Blanc, it was in Noir instead of Negro and Blanco. So I thought that would be sort of rude too. So that's how they sort of stuck together. And some people have been quite not upset, but disturbed by my jocular nature. But in fact, when I thought of those varieties, the goal was not necessarily to produce a varietal wine from that variety. The goal was to produce a blending variety that you could grow in PD hotspots and be done with it. But in fact, the wines are so nice they can easily stand on their own, and most everyone who's tried them agrees with that. But they're stuck with the name now. But they'll probably package them in some sort of a fanciful label and be done with them at that point.
Maybe in the not too distant future, the department will be able to sell wines made from your grapes. Wouldn't that be great?
Yeah.
Okay, so here's a semi-technical question, and I think you might guess who it came from. What two underrated grape varieties would you cross to create the next great wine grape variety?
This question came from, it may not have come this time from Randall Graham, but it did originally come from Randall Graham, and it launched a 3 or 4 year philosophical walk through viticulture and through ampelography. There is no good answer to it. Whatever you'll do will be interesting for sure. I don't think you have to, again, I personally don't think it's useful to breed new wine grape varieties. There's 3,000, that's a lot. What's important is to breed a disease-resistant variety and make sure that we're making some inroads on the huge need of sustainability and trying to reduce pesticide use and fungicide use. Those are really critical issues. And eventually, society is going to say to viticulture, we don't want you applying that much of these materials to control mildew diseases, and we're going to have to do something about that. And we don't have a lot of good choices in terms of fungicides that are organic. So it limits our palette of treatments. So clearly we should have disease-resistant, mildew-resistant vinifera varieties. And they could range from one style to another within that, but we need them desperately. And of course, we were partway to getting them. The French were in the 1860s, 70s, 80s with the French hybrids or the hybrid direct producers, which relied upon Riparia, unfortunately for them, because it's not necessarily a very good wine grape variety, relatively neutral, but very astringent and very acidic. And they didn't go through enough generations. They fell into the trap of making wine at each generation, which is a serious mistake. We have to consider that the only good ones are going to be made at the end of that cycle of multiple generations of backcrossing. And then we can accentuate both quality and resistance in a wine.
I have a friend in Europe who is planting, I'm not sure, I don't remember the names, but they are referred to as no spray varieties, right? So these are hybrids of some sort. And I think, he's a member of some club and these folks all, try different grapes that don't need sprays for exactly that reason. In other words, the goal here is to be able to cultivate grapes and make wine with very few inputs in the vineyard.
Yep. And I think that's a noble goal and a critical one. And it's been attempted from both a molecular genetic perspective and a more classical genetic perspective all over the world. Almost all the world's grape genetics labs are working on mildew resistance of some sort, either downy or powdery. It's good,
Right.
important.
So we have a question that came in on the Q&A, which is quite specific. The questioner asks, he heard a story that Carmenere became common in Chile, I think it says, because it was resistant to phylloxera. He wonders if this is true. This person really likes the flavor of Carmenere, which puts this person in an unusual classification, and wonder why it hasn't done well in the US. I can answer that part.
It's not that bad, but
If
it's
you have
not.
an opinion of the correct Carmenere story and why isn't there more of it in the US?
Yeah. So it's not phylloxera resistant. It's vinifera. There's an Olmo variety that's similar, Carmine, which is very, very terrible. It wasn't well done. I don't think Harold had much of a sense of pyrazines. So it's a real pyrazine bomb, if you like, if you like that characteristic. But the true Carmenere is from perhaps Torino and perhaps in France. But it's one of the interesting grapes that went into both Argentina and Chile before the phylloxera crisis in the 1820s and 1830s. They actually have a snapshot of what was done viticulturally all around Europe in the South American vineyards most often, because the plant material came in from that era. And it was before phylloxera, and after phylloxera wiped out a lot of those vineyards. So in many cases, Chile and Argentina have rare clonal and varietal forms of the given set of grape varieties, because they came in so early on, not later, not in a modern sense.
So the Carmenere is not resistant to phylloxera. It's just grown in a place that doesn't have phylloxera.
Right.
Yeah.
Not for long, maybe. Who knows? I've noticed they've stopped inviting me to Chile over the years.
So do you have an opinion on why Carmenere isn't more popular in the US?
Carmenere or Carmine?
Carmenere.
Carmenere, yeah, I don't know. And it's not that bad. It's not a bad grape. It was part of the blending sort of philosophy of that era of Bordeaux, for instance, and of northeastern Italy too. I don't know. It's a quick answer.
My experience is that the vegetal character is a little bit too strong.
Yep.
At least
it's stronger than Cabernet Sauvignon and Cabernet Franc for sure.
So here's one that's where is the DNA sequencing done? Not sure how to answer that.
Where's it done? Well,
Yeah.
we do some amount in our lab, and we utilize the technology in our lab, and it's done in other labs. Dario Cantu has a lot of sequencing going on, and that's mostly done in computers now. Once the machines generate the pieces of DNA, then they're all fit together like a giant jigsaw puzzle by computer. So it can be done privately. It can be done in a given lab, and there are services that offer it at various universities, including ours.
All right, so here's one that's a little more personal. Are you going to go back and improve your tennis game?
I haven't picked up a tennis racket. Well, it's not true. I haven't played good tennis in 50 years. So there you go. I guess I should start.
And here's one from someone in the Olmo family. Are you really old enough to retire? Dad,
Well, yeah,
thought 92 was a good age.
92, yeah. Yeah, he did pretty well. He was more patient than I was.
And this person says they like carnelian.
Carnelian, yeah. So another one of almost his last set of red wine grapes, Carnelian, Carmine, and Centurion, all are quite similar. And they're based on the same sorts of parents. One of the parents was a female flowered variety, so it's easier to use as a parent. It didn't have to be emasculated. Most of the cultivars are hermaphroditic or perfect flowered. You have to remove all the stamens before you can make the cross in that direction. But they're not bad. They've never really done very well. It's hard to introduce new grapes, incredibly difficult. If they were mildew resistant, they'd been snapped up. They'd have been used.
So this is I think maybe this is our last question. They're asking about resistance to phylloxera, and I guess this refers back maybe to XR1, questioning whether or not phylloxera resistance is going to break down. I'm not sure. And then is phylloxera going to continue to be an issue for the American wine industry?
That's an interesting question, and we wrote a paper up. Samara, in fact, spearheaded it, looking at about 60 different rootstock cultivars from both France and California. And we were looking to see how much diversity existed in those rootstocks, and diversity from the perspective of genetic breadth, not very narrow basis. So we wanted to look at the parents, and it turned out there are, of course, very few parents. We knew that already: the Riparia, rupestris, and Berlandieri, and that's a longer story, but we'll just accept it. That's the way it is. But the scary part was not that there are only 3 species in vast majority of all our rootstocks, was that the same accessions were used as parents. In other words, the same Riparia, exactly the same Riparia, was used in a very large number of crosses, exactly the same rupestris, and exactly the same Berlandieri. Now that's when you should get worried. Not so much about the phylloxera's ability to suddenly become super powered, but from the constant battle between the plant and the pest. Eventually, that may break down, and we don't have any alternatives. There's no diversity in that planting. So if phylloxera evolves to overcome those wild species that are in those selections of rootstocks, it'd be bad news. So we should be breeding more phylloxera-resistant rootstocks. That's Astrid's next career. You can start working on just that component of it.
All right. Well, let's get our panelists back here. Let's see if we can get them back.
They're all there.
They're there. Are they there? Here they come. All right. So I guess we'll go in the same order we did before, and ask Luigi if he's got any final comments.
Yes, I think that after this forum, everybody has realized that how crucial is the role of the breeder, how crucial is breeding to promote resilience instead of managing risks. Climate change has been described before. A question from Andy Waterhouse to Andy Walker. And concerning the retirement of Andy, I think that the retirement is like the passing of the torch in a relay race. And Andy will be for sure able to transfer the newcomer all the knowledge he has, the enthusiasm Andy has, and teaching this newcomer that finally the role of a breeder, the role of science is to benefit the society. The final role is that one, beyond any metrics which is nowadays important in the academia. So Andy, a good retirement, and take care and see you soon. And thank you for this very exciting forum, this event organized for you, because you deserve this. And bye-bye.
Yes, thank you, Luigi. Ciao.
Ciao.
I realized I left potentially, well, several different adjectives off of Andy, but one of them that I really wanted to mention is his passion. And what you can see is how contagious in the best way possible your passion is, and it's translated to all the people that have spoken tonight. And we're so forever grateful for everything that you've contributed to this industry. And still we're all in denial that you're retiring, but we wish the best for you and your family, and your legacy lives on. So thank you so much, Andy.
Thanks, Remy.
Astrid?
Yeah, I certainly agree with everything. And thank you for having the chance of being here. And thank you for that last question. That gives me the chance to skip in, because it's true, phylloxera won't disappear. And it will have to be, it is challenged by other stresses that we find in the root, in the soil, whatever you mentioned, Sumira, but also on the leaves. So we see it much more. So it will be fun to see what it's going to do on the new hybrids that we see. So besides of phylloxera, thank you so much also for giving us the chance to learn about everything, not only about breeding phylloxera and everything, but also about teaching. So thank you so much, and all the best to the walks, and lots of fun with your kids and grandkids.
Thank you, Astrid.
Smyra, go ahead.
I will continue the thought process which Astrid started. We don't know the climate change, how it's going to impact for the phylloxera and interaction of the plant and the pest. And I don't think it's going to go away. Maybe as the warming temperatures, I think that many new diseases, their zones are going to be extended, and breeders and the germplasms are always going to be the necessity. So wrapping up that thought, and I think that I am in denial. I believe that. I don't think Andy is retiring. And because we have a lot of stuff in the pipeline, we will still be testing, and we will be still releasing plant material, which is powdery mildew and PD resistant, and many other attributes. And I am looking forward to working with Andy and having him as my mentor when I start my own independent career. So thank you, Andy, for being there always when I needed guidance, when I needed some kind ears to listen to my frustration. And whenever I hit a roadblock in research, in my thought process, to just taking me out of my tunnel vision. And it has been a tremendous opportunity to work with you and work with everyone in your lab, and became so many new good friends. Thank you so much.
Thank you. Thanks, Samira. I didn't have a chance to thank anyone either. So thank all of you for coming. That's fantastic, particularly when you're 2 or 3 in the morning and your heads are getting heavier. But of course, I want to thank Viv, my wife Vivian, and my daughters, Hannah and Maya, and my new sons, James and Stephen, sons-in-law, and the 4 grandchildren who are now keeping me quite busy. Not as busy as Viv, but they're quite active. And there probably won't be time for tennis. That's the good news. I have to worry about hurting my back. But thank you so much for your very kind words and your ears and listening and years of interaction have been fantastic. Thank you.
I'm Eric Wada, one of Dr. Walker's former graduate students who worked on Systematics of Vitis, the grape genus. I'm honored to celebrate Dr. Walker's career and to answer the question, what is one thing Dr. Walker signifies for you? My answer is that Dr. Walker signifies resilience. Being a grad student, one can easily get beat down by missing out on a grant or a scholarship, having a paper rejected, troubleshooting experiments, etc. I remember my first collecting trip with Dr. Walker and another one of his grad students, Kristen Lowe. We took a red-eye flight to Texas, drove over 2,000 miles in under 72 hours, and came away with far fewer specimens than we had anticipated. We followed up by talking about ways to improve future collections and ways to move forward with the research, and of course, subsequent collecting trips were much more productive than that one. Through it all, through my graduate career, Dr. Walker was supportive, empathetic, and resourceful. I'm now a faculty member and soon to be Academic Senate President at Folsom Lake College, a primarily teaching institution that serves many underserved and underprivileged students, many of whom are the first in their families to go to college or who have had significant gaps in their educational journeys. I am inspired and continue to be inspired by Dr. Walker's resilience. I try to emulate his supportive and empathetic character to help my students succeed in their educational goals. Dr. Walker, Andy, thank you for supporting me both through my graduate education and my career choice.
All right, well, thank you all for coming this evening. I'm going to move on and close us out. So thanks again for coming. I'd like to mention that the department is honoring Andy Walker with a in-person program about a year off, May 19th, 2022. So put that on your calendar. Also, we do have a survey, an exit survey as you leave this event. But tonight we have a special question. If you want to leave any comments or thoughts for Dr. Walker, we'll pass those along to him. So please take an opportunity to leave your comments there. We have one upcoming event later this month. On Wednesday, March 26th, we'll host a Sips and Bites discussion on beer wine mashups. Professor Glenn Fox will host a discussion with two brewmasters from the brewery in San Diego. We do have samples that you can purchase to taste along with our guests. I'm not sure how long those will be available or if they still are, but we will have an interesting tasting. These are beer and wine hybrids. Finally, I'd like to give a call of thanks to our friends of the RMI for their support and help provide the means to provide programs like this to you. And finally, I hope you enjoyed tonight's presentation. Good evening.